Hello everyone. So today we discuss about receptors, nuclear family receptors, which is a type of receptors. At first, we discussed in the previous class that what are the types of signaling molecules. Signaling molecules is of two types. One is polar and another is non-polar. So polar molecules are hydrophilic in nature, that cannot or unable to cross plasma membrane, whereas non-polar molecules are hydrophobic in nature, that can cross plasma membrane. So those who can cross plasma membrane, they also cross nuclear membrane. So that's why nuclear family receptors are the receptors that are found in both cytosol or nucleus. Here you can see here present in both cytosol or in nucleus. So those receptors which are present in nucleus or cytosol both, they can accept only non-polar signals, as the non-polar signals can cross plasma membranes, but cannot accept polar signals because polar signals cannot cross the plasma membrane, so they can bind only surface receptors. So here nuclear family receptors are the types of one type of receptors mainly of various types, like GPCR, G protein coupled receptor, tyrosine kinase receptor, ligand gated ion channel receptors, and among them nuclear family receptors are one. So nuclear family receptors accepts or receives polar signals, non-polar signals, not polar signals, and they present in both cytosol or nucleus. They act as transcription factors. Every nuclear family receptors acts as transcription factors. So nuclear family receptors have a ligand binding region. Here you can see here. Ligand binding region, this is a ligand binding region, and the ligand molecule binds in the ligand binding region, and after then there is a DNA binding region, and this DNA binding region contains C4 zinc finger motifs. C4 zinc finger motifs can be discussed below in details. Then this C4 zinc finger motifs interact with DNA through hormone response elements. This also discussed below in details. And now this is the primary structure of nuclear family receptors. So like after the during cell interaction, ligands comes to the receptors, and the ligand binding domain, this position the ligand binds with the receptors, and then the signal passes through the DNA binding domain, where C4 zinc finger motifs interact with DNA through hormone response elements, and the mechanism gets started. Let's us discuss about zinc finger motifs. Here you can see zinc finger motifs. Zinc finger motifs has 4 cysteine molecules. Here 4 cysteines are adjoined with the zinc. This is called zinc finger motif. Zinc finger motif is a protein domain. As you can see here, these are amino acids. So it is a protein domain that controls gene expression by binding with DNA. It binds with DNA and it controls the gene expression along the mechanisms. And then we move on to the hormone response element. So this is the structure of hormone response element. You can see here hormone response element is a DNA sequence. Here is the DNA, and it is a sequence in DNA that binds to the hormone receptors to regulate gene expression. Here you can see that the target gene can be expressed when the ligand binds to the receptors, ligand binding region, and then hormone response element here is a sequence in DNA that binds to the hormones, and it allows the promoters to change their transcriptional rate in response to hormones, such as insulin receptor genes. Negative response element reduces the cell's ability to remove glucose from the blood. Just like these hormone elements works, and the target gene can be expressed. Now let's discuss about the detailed structure of nuclear family receptors. So this is a detailed structure of nuclear family receptors that you see here. Here you can see N. This N is amino groups that are attached to the receptor, and this A or AB is a variable region where activation factor 1 lies and walks. And this C region, this is domain C, and this domain C region contains DNA binding domain, which has zinc finger motifs. As I have told before, that zinc finger motifs is a protein domains that have 4 cysteine molecules and interact with DNA along with hormonal response element. D domain has hinge region and NLS, nuclear localization signal. So what is nuclear localization signal? Nuclear localization signal is an amino acid sequence that tags a protein, that protein which is synthesized in the cytosol, and the protein is imported to the nucleus by nuclear transport. So NLS is a type of identity card that helps to cross the barrier and permits the protein to enter into the nucleus. That is NLS. E region is ligand dependent dimerization domain where co-repressor binds, co-activator binds, and it is a site of dimerization where nuclear family receptors are dimerizes. So co-repressor when binds to the ligand to the receptors, mainly in the ligand binding site, then it inactive the receptor. But when co-activator binds, it will active the receptor. So this will discussed further in the mechanisms. You can see here C is binding. So C is carboxyl groups. Here amino acid groups, amino groups, and this is carboxyl groups. Now we discuss about the types of the nuclear family receptors. So you can see here that nuclear family receptors is of two types. One is type 1 that is present in cytosol, and another is type 2 that is present in nucleus. So it can be divided in the presence of the receptors where the receptors are present. In present, if it is present in cytosol, then it is type 1, and if it is present in nucleus, then it is type 2. And the receptor present in cytosol are acts as homodimer, whereas nucleus receptor present in nucleus are acts as heterodimer. And the receptor bound with HSP90 in cytosol, and in type 2 receptors bound to the HRE, hormone response element, in nucleus. When it bounds with HSP90, it will in inactive state, and when it binds to the hormone response element, then it will be activated, and target gene expression will be expressed. Here are some signal molecules, signal examples that are discussed. Testosterone, estrogen, progesterone, glucocorticoid, mineralocorticoid. These are the examples of type 1 receptors. And retinoic acid, thyroxine, 125 dihydroxycholecalciferol is an example of type 2 receptors. So in this part, up to this, and in the next part, we will discuss about the receptor signaling. First type 1 receptor signaling, and another is type 2 receptor signaling with examples.